Hello everyone, my name is Hitoshi Nakatogawa. I'm an associate professor at School of Life Science and Technology at Tokyo Tech. I'm glad to take this opportunity to introduce my research. So my research theme is Unravelling the Mystery of Autophagy. And the cell is the most fundamental structural unit of life. And our body is an assembly of about 600 trillion cells. And autophagy is a phenomenon that occurs within cells and known to be triggered by different signals, such as nutrient starvation. When cells receive these signals, small flattened membranes appear within cells. And these membranes expand while curving and finally close to form spherical double membrane sacs or vesicles called autophagosomes. In this process, various cellular components are sequestered into autophagosomes. The autophagosome then fuses with a lysosome in mammalian cells or vacuoles in yeast and plant cells. And because lysosomes and vacuoles contain different degradation enzymes, the contents of the autophagosomes are degraded by these enzymes after fusion with these organelles. We can often compare the autophagosome to a garbage truck that collects and transports waste, while the lysosome vacuole can be regarded as a disassembly or recycling plant. There are two types of autophagy called non-selective autophagy and selective autophagy. Non-selective autophagy is strongly induced by nutrient starvation, and cytoplasmic components are randomly sequestered within the autophagosome and degraded in lysosomes or vacuoles. And the degradation products are used as nutrients for cells to survive starvation conditions. In selective autophagy, specific targets such as disease-related abnormal proteins or organelles are selectively engulfed within the autophagosome for their elimination in lysosomes or vacuoles. In this way, two types of autophagy degrade a wide range of cellular components under different conditions to maintain the health of our cells, therefore our bodies. Indeed, recent studies have revealed that autophagy is involved in many physiological processes, including starvation response, anti-aging processes, the presentation of antibodies, development and differentiation, organelle homeostasis. In addition, the failure of autophagy is suggested to be linked to a number of human diseases, including neurodegeneration, heart failure, diabetes, liver disease, microbial infection, and cancer. So this great expansion of the field of autophagy research was triggered by the discovery of genes required for autophagy in baker's yeast by Dr. Yoshinori Osumi. And he won the Nobel Prize in Physiology or Medicine in 2016. I studied autophagy research in his group in 2004 and had my own laboratory here in 2017. And my work is working on fundamental issues about autophagy using baker's yeast Saccharomyces cerevisiae, including the molecular mechanism of autophagosome formation and what are degraded by autophagy, and how the selectivity is determined, and what is the physiological significance of their degradation. And here, among our previous achievements, I'd like to introduce our recent finding on selective autophagy of the nucleus and the endoplasmic reticulum, ER. This slide shows the mechanism of target recognition during selective autophagy conserved from yeast to humans. And targets of selective autophagy are first recognized by proteins called autophagy receptors, and these receptors bind to a protein called ATG8 on forming autophagocytal membrane, resulting in the efficient sequestration of the target into the alphagosome. At that time, in Saccharomyces cerevisiae, these proteins and organelles were known to be targets of selective autophagy, and autophagy receptors for these targets were also identified. But we assume that there still remain unknown targets and receptors for selective autophagy, and given the fact that these previously reported autophagy receptors all interacted with ATG8, we aimed at identifying novel autophagy receptors among proteins interacting with this protein. Actually, we isolated ATG8 from yeast cells, identified proteins isolated together with ATG8 by mass spectrometry, and focused on two proteins nobody had analyzed before, which we named AT39 and ATG40. Analysis of these proteins led us to discover selective autophagy of the nucleus and endoplasmic reticulum. Both of these proteins are expressed in response to nutrient starvation, and ATG39 and ATG40 localized to the nuclear envelope and the endoplasmic reticulum membrane, respectively. And both interact with ATG8 on forming autophagocytal membrane to efficiently pack the fragments of these organelles into alphagosome for degradation in vacuoles. We also found that yeast cells are deficient for selective autophagy of the nucleus or nucleophagy die earlier than wild-type cells during nutrient starvation, demonstrating the physiological significance of nucleophagy in the maintenance of cellular homeostasis under nutrient starvation. About selective autophagy of the ER or erophage, at the same time as our identification of ATG40, Dr. Ivan Dekic's group in Germany discovered FAM134B as a receptor for erophage in mammals. Since then, several erophage receptors have been discovered in quick succession in mammals, and some of them were suggested to be associated with human diseases. We were very happy that our research in yeast could largely contribute to the discovery and understanding of erophage. Finally, I'd like to emphasize that autophagy won the Nobel Prize, but many mysteries still remain to be unraveled. I didn't talk about molecular mechanisms underlying autophagosome formation this time, but many interesting issues remain to be clarified. In addition, we don't yet have the whole picture of autophagic degradation, what are degraded by autophagy, and when and how much they are degraded. And the relationships between autophagy and the physiological phenomena and diseases are also still unclear. We need to understand them at a molecular level to control these phenomena and develop drugs to cure these diseases. Thank you for watching. If you like to know more about my research, please check out the links in the description box.